So Kim gave us an overview. Betty talked about cyanobacteria in streams in California, and the next one's going to be a specific river, the Eel River, and the ecology of cyanobacteria and toxins. It's the presenter is Keith Bauman. That's right, like Bauman Gregson, and he's a PhD student at Berkeley, and he's going to come and talk about his study.
Thanks, Jim, for inviting me to speak here. And my name is Keith. I'm a PhD student at Berkeley, and today I'll be talking about cyanobacteria in the Eel River in Northern California. And the Eel River is a 10,000 square kilometer watershed, and it flows through the coastal range of Humboldt and Mendocino counties. And on the mouth is up here, which is just south of Eureka and Arcata. And being in California's Mediterranean climate, we've got really high discharges in the wintertime, but in the summer, when we get the drought, the width of the channel recedes quite extensively. You get really clear water, warm temperatures, and this is a prime environment for growing lots of algae, which you can see here in this photograph. And this is really important for the ecology because the algae forms the base of the aquatic food web during the summertime. And my advisor, Mary Power, has spent a lot of time researching this food web. And what she has found is that the basic process is that in the spring and summer, we get these large mats of green algae, primarily Cladophora, will grow up in the river. And then over time, these will get epiphytized by diatoms, which are single-celled algae. And you can see this in this SEM photograph, this electron micrograph photo. And diatoms are really nutritious, highly edible, and so the insects and the fish love this stuff. And over the course of the summer, you get a really vibrant and productive food web with insects and fish consuming algae, which then also are consumed by birds and bats as the insects emerge from the river, creating these really productive river corridors. However, it's also been documented that the algae can kill dogs in the Eel River. And when I found this out, this really struck me as surprising because I had just finished a master's degree at the University of Michigan. And in the Midwest, when people talk about cyanobacterial blooms, it's usually in a lake, like what happened in Lake Erie this summer. It usually relates to agricultural land use and nutrient runoff. And so to be having harmful algal blooms and dog deaths from cyanobacteria happening in a river, first of all, that's flowing through relatively low population density, no large corn agriculture, not a lot of nutrient runoff, I was like, this isn't the textbook case of cyanobacterial blooms that you learn about in your limnology classes. And so I decided to set out and try to figure out, well, what's causing these algal assemblages to tip towards toxic cyanobacteria? And in this slide and the previous slide, what we're seeing here is all these darker vertical spires are mucilaginous cyanobacteria that's epiphytizing the green algae. So instead of having diatoms on the green algae, we're getting cyanobacteria. And these can create large mats that can become the dominant paraphyte in the river. So what might be some of the consequences of this? Well, first of all, we could have effects to the aquatic food web and our secondary production. So the diatoms in the green algae, really edible. We've documented that it supports a lot of secondary production. The cyanobacteria, as Kim and Betty also mentioned, organisms don't like to consume them. They've got all these active metabolites, and so you could find that you're not able to move as much primary production into secondary production, and so you won't be able to support as many fish and insects. And then you also have all your public health concerns, where the toxins create public health threats. Already, Humboldt County posts warnings about accessing the river in the summertime. There's been over 11 dog deaths in the last decade attributed to cyanotoxins in the river. And so what I'll be mainly focusing on in this talk are microcystin toxins and antitoxin A. And that's because microcystins are the most well-researched in the literature. But in the eel, most of the dog deaths were attributed to antitoxin A, which is a neurotoxin. But the data actually from necropsies or algal samples was pretty scant over the last 10 years. And so I was trying to get a better understanding of what are the toxin dynamics. So Kim and Betty mentioned this a bit, but some of the factors that are associated with these cyanobacteria blooms, well, first of all, most of this is coming from the lake and estuary literature. Less of it's been researched in rivers, especially with the benthic cyanobacteria. But cyanobacteria do better at warm temperatures. They can scavenge nutrients really well. They can fix nitrogen. There's been some work to show that some of the species are very tolerant of ultraviolet radiation, and they do well when carbon levels get low and pH gets high. But one of the really important things that's unique and needs consideration in the rivers is the effect of flow, because flow serves as a master variable. All these other things, temperature, nutrient and carbon delivery, dispersal, drag, are all a function of the flow environment that the algae is experiencing. So it could be possible that as the flow regime shifts, you might be, you're going to affect all these other environmental dynamics which could favor cyanobacteria. Now, as I mentioned before, the eel is this large 10,000 square kilometer watershed, and I knew that I couldn't sort of fully document what was going on there all by myself as just one lone graduate student driving a minivan around. And so I was really fortunate because starting with the PhD, I was really into and passionate about scientific outreach and citizen science. And the year before I began my program, this group called the Eel River Recovery Project had started in the watershed. And they had really gelled and sort of gained momentum around the good news of increasing salmonid returns in the watershed and the troubling news of increasing cyanobacterial abundance in dog deaths. And so I've been working with them for the last couple of years to help do training events. So we'll put out some press releases, try to get citizens down to the river. I'll do workshops on how to identify the cyanobacteria, how to make sure that people know what to avoid when they're down at the river. We also, Mary, my advisor, we have a field station at the Angelo Reserve, and so we hold an algal workshop every couple of years. We invited a bunch of citizen scientists down to teach them how to identify algae with microscopes. And so we're slowly developing this grassroots network in the watershed of people who are equipped to identify cyanobacteria at the macroscopic level, just out in the field, but also at the microscopic level. Now, all of this is not that formalized, but we're trying to slowly to move this in a direction where we can get better protocols, more formalized monitoring within the watershed. And then it's also been great for me because people have been very generous, giving me access to their private property to collect samples from the river. I've been training people on collecting samples, and so I'm really indebted to all the citizens in the eel to helping me collect the data that I'm about to present here in a moment. And so the general driving question for the last 2 summers has been, what is the temporal and spatial distribution of the cyanobacteria in the Eel River? Like I said, with 10 dog deaths over the last decade, nobody really knew where and when the cyanobacteria was growing. And so we set up a bunch of monitoring sites that were visited weekly over the course of the summer. There was, these are the sites from this last year, but the first year I only had 7, and this year I now have 10. We collected algal samples, we were measuring cyanotoxin concentrations, had temperature probes in there, and we're also collecting water samples for total dissolved nitrogen and total dissolved phosphorus. And all of these sites here were where I had volunteers collecting samples for me. And also to orient yourself one more time, this Angelo Coast Range Reserve is a part of the University of California Natural Reserve System, and that's home base for the summer, and it's in Mendocino County. So what did we find? We now have a much better handle on what cyanobacteria is really growing in the river and causing these large benthic mats. What's primarily epiphytizing the green algae are species of Anabina and Cylindrospermum, and then occasionally you'll also see low abundances of nodularia in there as well. And there's several different species within each of these genera. I'm working on keying those out a little bit further. I'm a little bit backlogged in my microscopy, but I have a bunch of samples that we're working through with some undergrads right now, so I'm hoping over the next couple months to be able to add a little more resolution to this. Also to note, all 3 of these can fix nitrogen. There's another dynamic that I didn't show you many photos of, but there's also Formidium mats. And Formidium is another taxa that, but this one does not fix nitrogen. And it lives in the faster flowing water and creates these really pretty and kind of crazy looking brown and orange felty mats on the fast cobbles. And in New Zealand, they've been having really big issues with Formidium mats in their rivers, and that's one of the main producers of cyanotoxins in New Zealand. And we get these growing in the riffles. And then here's the nostoc that Betty was just talking about. It creates these really three-dimensional, thick mucilaginous balls. And this has been present in the river. And I owe Betty a big thank you because there was some conversations earlier this year with her that got me thinking more about nostoc, because usually it's not researched as much, and I had kind of written it off as not a toxin producer, but I'll show you in a little bit. We did document that it was is producing toxins up in the watershed. So our understanding of what taxa are there is increasing, and I still have more samples to go through, so it's exciting to watch the patterns come together. To measure the cyanotoxin concentrations, I use this method called solid phase adsorption tracking sampler, which is a mouthful, so we'll just call it SPAT samplers, and Betty also just mentioned these. And so these are this really neat passive monitoring method where you place these rings of this HP20 diion resin into the river, and any toxin molecules in the water column will absorb onto this resin. And so what I was doing was putting them in the river for 1 week, then we pull it out of the river, extract all the absorbed toxins that were stuck on the ring, pull them off the ring, and then we would measure it with liquid chromatography mass spec. And we were doing that for microcystin and anatoxin A. And what's nice about this is rather than getting a grab sample where you just stick an Nalgene bottle in and get this instantaneous snapshot of one little parcel of water, here you're really getting a better understanding of what was happening over the course of a week in that river. And here's a couple publications coming out of the lab at UC Santa Cruz, Professor Cadella's lab, that has really been promoting this method in the state of California. And so I want to just show you how this data is then visualized, because it's a little bit different from the micrograms per liter, which often the data comes in. So you get units of nanograms of toxin per milligram of resin per day. And so each one of these horizontal dark lines is one sample ring. And then you get the average daily toxin concentration. And then when I put a new ring in, you get a new value for that week. And then you just can create this neat time series of what was happening. And we're not really trying to estimate the flux of toxins downstream. Rather, what this is giving us is this relative time series. So I think of it as something akin to a catch per unit effort in a fisheries, for fisheries uses. So I want to make sure that's understood. So now I will show you some data. And I'm very excited because I just got the 2014 results on Monday. So I've been frantically scrambling the last 24 hours, 48 hours to make some plots, which is exciting. But here's 2013. Some of you may have seen this already. But what was exciting about this, the first thing to notice is the axes. You'll see that the ranges are different, and we had a lot more anatoxin A in the water column. So this was exciting because it confirmed a lot of those anecdotal stories or the scant data that was linking the dog deaths to anatoxin A. Well, now we have better spatial and temporal data to suggest that anatoxin A is more prevalent. Microcystin is prevalent, and was intriguing here was most of it was coming at the Angelo Reserve, which is our field station, which is high in the watershed, was a part of the Nature Conservancy since the 1950s, and the UC Reserve in the 90s. So it's usually thought of as a relatively pristine and reference site. So we were curious, maybe there's just a natural amount of microcystin that could be being produced in these rivers, but not really sure about that. As for the anatoxin A, most of the production was happening at these three sites, and these are all on the South Fork of the Eel River. And if any of you have been up there, Highway 101 eventually parallels the Eel River once you get up into Mendocino County. And so these see a lot more recreation use, a lot more developed in terms of land use. And also there have been a lot more dog deaths in this region as well. And then the timing of this is that things are peaking end of July into mid-August. So that was 2013. Now, for 2014, here's just the microcystin data. And this is only about 3/4 the samples. I didn't get it all finalized. But a similar dynamic where Angelo is the site that has most of the microcystin. Numbers are a little bit higher than the previous year, but other sites don't really seem to be producing it. So this was encouraging to see similar trends in both years. It's likely that this is all real data. This is reflecting what's going on in the river. As for the anatoxin A, the same sites, PV, RG, and SH, which are those sites on the middle reaches of the South Fork River along the 101, are the ones that are spiking up again around the same time, end of July into mid-August. And I actually only have about 1/3 of the anatoxin A results here, so we'll be getting a lot more of that. This was exciting and encouraging to see similar trends in both years. I feel like we're starting to get a handle on what's happening both temporally and spatially in the river out of this SPAT data. Now, another thing that we did this year was we actually took scrapings of algae to see, okay, well, we know what's happening in the water column, but what are the actual algae that are growing on the cobbles and the benthos doing? And here's a plot of that, or a table for this. And I only have 10 of these samples, but I have about 110, 120 total. We just haven't processed them all yet. Here's the column for just presence, detection of microcystins, or anatoxins. And you'll notice that the formidium, some samples were just producing anatoxins, some were producing both, but we're hoping that after the whole 100 samples is collected, we'll have a pretty good sense of what the major players are in the river. And here's those nostoc samples that Betty's curious about, and we found them, both of them, to be producing anatoxin A, and I've got more samples to come. And then the other goal is to put this into terms of grams of toxin per grams of dry weight of biomass, because this is what those California OEHA action levels are stated in. And when I just looked at this, it was pretty crazy because the acute toxicity for a dog for anatoxin A is 0.3 nanograms per gram, and that's like down here. And we're getting ones that are, this is both over 200 nanograms per gram, this is 28 and 7, so pretty high. I do want to mention a caveat. I want to go through and once we get all 100, see if this is really real, just because of how high these values are based on the action levels, because this would really mean we have some very, very toxic benthic mats that are, real hotspots. So I'll be curious to see once we get the rest of the numbers in and maybe rerun some samples, how things pan out. So then the other big goal is to try and correlate this to some of these environmental drivers. So we had temperature data. This is all arranged from downstream to upstream. It gets warmer as you go, get to these middle reaches, PV, RG, and SH, where most of the production was. These two here are on a different stem, so that's why they're a little bit separate. As far as nutrients, we have a lot of nitrogen fixation in the river, so these dissolved nitrogen stay relatively constant. N to P ratios drop over the course of the summer, so you're getting a change. P drops, so your N to P ratios are changing. And so what I'm starting to do now that I have this data is look at these relationships between the environmental variables and the toxin concentrations. Here's a quick example of that, where we've got the N to P ratio on the x-axis and the toxin concentration on the y-axis. And we're seeing that it's like relatively intermediate N to P ratios where a lot of the production is happening. And this is just the 2013 data, so I think with the 2014, we'll flesh this out a little bit more. And so here's some of the hypotheses that we're going to be testing next year. So now that we've got a better understanding of these spatial and temporal dynamics, next summer at the Angelo Reserve, I want to do some manipulative experiments looking at flow and nutrients to see what environmental drivers are affecting the growth rates and distribution of the cyanobacteria. I think flow, decreased summer base flows could be influencing temperature, which could all influence cyanobacterial growth. We know that when grazers get exported due to high winter flooding, that increases the green algal biomass. So I'm moving into a stage where we're now looking for testable hypotheses. And so these are, I want to acknowledge these people. I've gotten funding from EPA STAR Fellowship, NSF, some local graduate research grants, a huge amount of help from Mary and Professor Cadella, and a lot of the Eel River recovery volunteers and countless lab members. And so thank you for your attention. I'm happy to take any questions if there's time.
The Eel River runs through the Emerald Triangle.
Yes.
And I'm wondering if you've taken marijuana grows into consideration for the nutrients.
Right. It's something we're all aware about. I haven't, because it's not a well-documented industry and we don't have a lot of data on it, but I've come in close contact with it. So it's something that I'm aware of, and I think any drastic shifts in nutrient loadings and things across the range are probably attributed to marijuana. But it's also connected, I think, to road development with erosion rates, because a lot of the changes in the land use there are coming from the marijuana growth, but people need roads, which increases erosion, which increases particles in the river. But I'm not like fully actively trying to collect data on amount grown or nutrient runoff from some gross sites or anything.
We've seen a lot of pictures of the nostoc, the green ears that grow on the side of cobbles. Nobody's mentioned very interesting mutual symbiosis there, right?
Yeah.
The midge that lives
Yep.
in algae. The point I'd like to make here is some years ago there was Amelia Ward at Oregon State did some studies on the carbon fixation rates there and nitrogen fixation as well, and showed that it was very much enhanced by the presence of the midge. I'm wondering about the microsystems produced by the nostoc. Could that possibly also be enhanced? Thanks.
Yeah, that's a good question, and I did document when I collected nostoc. So for those of you not aware, nostoc's pretty neat. It has a symbiotic relationship with this little midge, where the midge will live inside the gelatinous sheets, and it changes the morphology of the nostoc. And I was documenting when I collected nostoc whether or not it had a midge present. And for a few samples, I also removed the midges, and I would like to do some testing of the biomass of the midge. So I might have some pilot data once I get through all my samples.
Yeah, I had a question back here.
Yes.
Yeah, so for your time series sampling, did you see at any point if there was a daily cycle for peak release of the cyanotoxin or reduction?
No, yeah, my resolution wasn't that tight. So I was just returning every week and not really sampling on an hourly basis or anything like that. But that would be interesting.
That was a great talk, thanks. I was wondering about so when the diatoms are present or appetizing the mats or the cyanobacteria, I mean, is that kind of widespread? Have you documented does it happen like on a yearly basis, like all of them are overtaken by the cyanobacteria, and then in other times it's diatoms? And then also is drought, a factor in all of this as well?
Yeah, so the general timing of things is usually as the green algae is growing up, the diatoms are starting to colonize it pretty quickly. It seems like the cyanobacteria are like the last stage of that. So it's usually you're finding like more aged green algae that's then getting colonized by cyanobacteria. So I'm curious if there might be the algae is like releasing a lot of nutrients, and maybe that's where the cyanobacteria is getting it from. But pretty much, and you still do see diatoms in those assemblages, and the cyanobacteria is patchy, so sometimes it'll be small patches that are connected, sometimes it'll be several square meters. So it's been difficult to fully always generalize it due to that patchiness. Did you have one more question?
Just about drought.
Oh yeah, drought. Yeah, so that's what I mentioned with the flows. I think it could be likely, with slower flow. The eel sort of becomes more pond-like with just little riffles and really a flowing river.
Just one last question.
Okay, so to the extent that N to P ratio can influence whether you switch on or off nitrogen fixation, it did look like along your N to P gradient you had a sort of, it looked like there was a response in terms of cyanotoxin production. I can't remember which toxin it was.
It was the antitoxin.
Okay. Do you think that there is a relationship, or do you have any ideas about whether you might do some studies seeing whether toxin production was related to whether or not nitrogen fixation is turned on? Because they're both energetically intensive processes. I can see them on the one hand potentially competing with another, but I could also see it looked like maybe the opposite was actually going on based on your data.
Right. Yeah, so yes, when I was mentioning the experiments, one of the first ones I want to try is looking at putting out substrates that can alter the nutrient concentration in the river and try to relate that to growth and toxicity, and looking at manipulating both the total concentration of nutrients but also the ratios, because there's been some pretty interesting research. There's some big data sets that have come out, like the big US EPA lake assessment and some Canadian lakes that have been trying to look at N to P ratios production, so yes, basically,
But
the short answer.
in the light of nitrogen fixation?
Yeah, on the light, yeah, I don't, it might be harder because that's harder to measure. I'm not sure if I could fully measure that. I'd have to see, but yeah, I think that there are definitely some trade-offs energetically, so, great. All right, thank you, everybody.
If there's more questions, very good, thank you.